Good afternoon, everyone. My team and I represent a novel 3 microRNA signature identified using bioinformatics predict survival in esophageal carcinoma. The significance of the research. Esophageal carcinoma, ESCA, which we will refer to as ESCA during the presentation, is characterized by aggressive malignant tumor formation. It is the 8th most common type of cancer and the 6th most frequent cause of cancer-related death globally. MicroRNA is one type of non-coding RNA which regulates cellular proliferation, migration, and invasion by modifying gene expression. In this research paper, differentially expressed microRNAs, which we refer to as DAMs, can be used as biomarkers for early tumor diagnosis, therapeutic strategy selection, and prognosis of ESCA. The method. First, they combine two microarray profiling dataset from the Gene Expression Omnibus database and RNA sequence from Cancer Genome Atlas in order to identify DAMs in esophageal carcinoma tissues. Then, data from 168 ESCA patients and 30 normal esophageal samples were obtained from CGA to assess the prognostic role of DAMs. After understanding that, they used the target scan to identify and predict microRNA target genes. Finally, further analyses were conducted using the database for annotation, visualization, and integrated discovery and protein P interaction networks were obtained using the search tool for the retrieval of interaction gene database, which also called STRING. Bioinformatics tools. Those are all the software they use to create the 3 microRNAs. And we add them the link so we can visit them because we are all bioinformatics students. So if you want to know more about the software they use in this research paper, you can click on the link and you will be able to see them. Analysis software and data bank. They use SPSS to analyze the result. They also use PRIMES, chi-square test, and student t-test, and also use a 5% significance. The data source. They use data from the Gene Expression Omnibus and the Cancer Genome Atlas. The 3 microRNAs form. Those are the 3 microRNAs that the researchers design using all the software tools that we presented in the first slide. Target gene prediction of it differentially expresses microRNA. Target gene were predicted using the microRDB, target scan, microRWalk, and Diana. The result of the research. The GEO microarray and CGA RNA sequence count dataset identified 27 upregulated and downregulated microRNAs. The Kaplan analysis indicated that there were 3 microRNA signatures significantly associated with the overall survival of ESCA patients. Target scan analysis show that the 3 microRNA signatures were a potential prognostic factor in ESCA. It was found that the target gene of the microRNA participate in various cancer-related pathways, especially the signaling pathways. More detail on the result. Univariate and multivariate Cox regression analysis showed that the 3 microRNA signature was a potential prognostic factor in ESCA. And Kaplan-Meier survival analysis identified the 3 microRNA signature to be microRNA 1301-3P, microRNA 431-5P, and microRNA 769-5P was significantly associated with survival of ESCA patients. And then gene functional enrichment analysis revealed that the 3 microRNAs participate in various cancer-related pathways, including viral carcinogenesis, Fockhead box-O, which they refer to as FOXO, vascular endothelial growth factor, human epidermal growth factor, and mammalian target of rapamycin, signaling the pathways. Limitation of the research. First, although the identified DAMs were associated with ESCA, No additional experiments were conducted to validate those findings. Second, although TCGA data validation was added to increase the sample size and confirm the findings, the majority of the ESCA samples were from esophageal squamous cell carcinoma. Therefore, further experiment exploration is needed to verify the ability of DEMS to determine the prognosis of ESCA. In conclusion, which is also key takeaway for this presentation, a promising novel 3 microRNA signature was identified as a potential prognostic biomarker for ESCA patients. More experimental studies with larger sample size are needed to validate the findings, and the mechanism of the 3 DAMs in esophageal cancer need to be elucidated. This is the reference, and then you can read the whole paper and see the image created with the different software. Thank you for watching my presentation, and let me know if you have any questions. Thank you.